Hello, my dear students. Welcome to Sankalp Learning Classes. I'm Vinuta Bhatt, working as a biology lecturer in Sankalp Learning Solution. Okay, so this is the first CET class. So that is on the chapter Sexual Reproduction in Flowering Plants. We are discussing the CET points related to the chapter Sexual Reproduction in Flowering Plants. Okay, yes. In today's session, I hope ಎಲ್ಲರೂ ರೆಡಿ ಇದ್ದೀರಾ ಅನ್ಕೊಳ್ತೇನೆ. Okay, so be ready with your notebook and pen. Please note down the important points so that will be helpful for your exam. ಸೊ ಇವತ್ತಿನ ಸೆಷನ್ ಅಲ್ಲಿ ಮೊದಲನೇದಾಗಿ ನಾವು ಸಿನಾಪ್ಸಿಸ್ ಅಂದ್ರೆ ಸ್ಟಾರ್ಟಿಂಗ್ ಅಲ್ಲಿ ಈ ಒಂದು ಚಾಪ್ಟರ್ ಅನ್ನ ಬ್ರೀಫ್ ಆಗಿ ನಾವು ಸ್ಟಡಿ ಮಾಡ್ಕೊಳ್ಳೋಣ. ಆಫ್ಟರ್ ದಟ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಸಾಲ್ವ್ ದ ಸಮ್ CET ರಿಲೇಟೆಡ್ ಕ್ವಶನ್ಸ್. ಓಕೆ, ಎಸ್, ಲೆಟ್ ಅಸ್ ಸ್ಟಾರ್ಟ್ ದ ಚಾಪ್ಟರ್. So flower, so mainly angiosperms means they are the flowering plants, mainly flower that includes the accessory whorls and the necessary whorls. Necessary whorls means male and female reproductive structures, androecium and the gynoecium, and the accessory whorls means petals and the sepals. So this is the structure of the flower that you have studied. Then here, the flower that is formed on the floral primordium, and the flower mainly that contain the androecium and the gynoecium. Androecium means that is the male reproductive structure, and the gynoecium that is the female reproductive structure. And so this androecium that produces the male gametes, and the gynoecium that helps to produce the female gametes. So let us see. This is the flower.
In this video, we will learn the parts of a
So the parts of the flower that you learned in your theory class. So I'm not going to explain the parts of the flower, so that you already know what are all the parts of the flower. So androecium, gynoecium, and the sepals and the petals, right? Here, mainly we are discussing the male reproductive structure, that is the stamen. So here, stamen, so that constitute the anther and the filament. Anther and filament together they form the stamen. Here, this anther, so that is bilobed, means two lobes are there. Here it is called as the bilobed and the dithecous anther. So very important thing about the anther, that is the bilobed and dithecous. So what is the meaning of dithecous? So bilobed and dithecous anther. So two lobes are there. Lobes, you know, two lobes are there. So when you take the section of the anther, you find so two theca. So this is one theca and this is the other second theca. So it is called as the dithecous anther. Okay. So it contains four microsporangia at the corners of this theca, right? So here four corners are there like this. So each theca, so dithecous, right? Each theca that contain two microsporangia, and totally four microsporangia are present in the anther, right? So that is called as tetrasporangia. Tetra means four. Here, these microsporangia, they grow into pollen sacs. Pollen sacs means they produces the pollen grains later. Okay, so that is the meaning of the dithecous bilobed anther, and this microsporangia that develop into a pollen sacs. Then the structure of the microsporangium. If you take the section of the anther like this, so this is the anther. When you take the section of anther, you will find microsporangia, right? So structure of the microsporangium. So that produces the pollen grains. Mainly it constitutes the four layers: epidermis, endothelium, middle layer and the tapetum. So these are the very important four layers. This is the epidermis outermost, and endothelium, middle layer, and this is the tapetum. Here this tapetum, that is the innermost layer, and the which provide the nutrition to the developing pollen grains. Pollen grains are produced inside this microsporangia and it provide the nutrition to the pollen grains, and remaining one, so they are protective in nature. So these are the four layers of the microsporangium. Then microsporogenesis. Yes, this is the process of the formation of the microspores. Right. Here, microspores are formed within the microsporangia. Here, within the microsporangia, there is a pollen mother cell. So, always remember, mother cell means that is diploid in condition, that is 2n in condition. So, which undergo the meiotic cell division. So, you know that meiotic cell division, that is the equational or the reductional cell division. That is the reductional cell division. The number of chromosomes that will become half, right? So the meiotic cell division and form the microspore tetrads, means the four microspores are held together, so they are haploid in nature. And these four microspores, so when they mature, when the anther mature, so these microspores, they dehydrate and dissociate from each other, and the microspores, they develop into pollen grain. Okay, so pollen mother cell diploid in condition, it produces the microspores, that is in tetrad condition. of During the dehiscence of the anther, these microspores, they develop into pollen grains, they dissociate and they develop into pollen grain. Pollen grain, that is the female male gametophyte. It is called as the gametophyte, not gamete. Pollen grain is the male gametophyte. And what is the structure of the pollen grain? Pollen grain mainly it contain the two layers, which contain the two layers: outer exine made up of sporopollenin and inner intine layer that is made up of the cellulose and the pectin, right? So this sporopollenin is the very hard structure. Here this is the hard structure. Sporopollenin is the hard structure, which is the most resistant material known, right? So here no enzymes and alkali, so that is not able to degrade this one. Okay, so that is the sporopollenin. Here where the sporopollenin is absent, so from that region, so it forms the pollen tube. It forms the pollen tube, and which is able to release the which is able to form the pollen tube and forms the some which is able to release the gametes. Okay, so that is the uh, germ pore where the sporopollen is absent. So here you can see, so this is the outer layer exine. In some regions the sporopollen is absent. So mainly this pollen grain, that is made up of the two cells, that is vegetative cell and the generative cell. Vegetative cell, that is the larger cell, has abundant reserve food and irregularly shaped nucleus. So this is the vegetative cell, and the generative cell is small, floats in the cytoplasm of the vegetative cell. So this is the spindle shaped cell. It is In over 60% of angiosperm, pollen grains shed at two-cell stage. So the pollen grains, they released at the two-celled stage, that is called as the vegetative cell and the generative cell. And remaining 40%, they divide mitotically and give rise to the three-celled stage. The gametes are formed. These generative cells, mainly they forms the gametes. So the pollen grains, they released at the three-celled stage, where the male gametes are formed before the release of the pollen grain. That is the three-celled stage. Two-celled stage means vegetative cell and the generative cell. Three-celled stage means generative cell that produces the two male gametes before the shedding. Okay, that is called as the three shelled uh, three-celled stage. Then what is the economic importance of the pollen grains? Pollen grains, they cause the disorders like asthma and bronchitis, and in foreign countries it is used as a food supplement, okay, and which is high in nutrients. Here, uh, what is the viability of the pollen grain? So that depends on the different species, okay. Here, in some cereals such rice and wheat, pollen grains lose viability within the 3 minutes of 30 minutes, sorry, 30 minutes of their release. So, and in some members of Rosaceae, Leguminosae and Solanaceae, they maintain the viability for months. Remember, some lose the viability of the pollen grains within a 30 minute, and some so that maintained about 30 about 3 months. For example, Solanaceae, Rosaceae and the Leguminosae family. And it is possible to store the pollen grains in the liquid nitrogen in minus 196. This is minus 196 degree Celsius. So that is in the liquid nitrogen. This temperature is important, and where it is stored, that is at the liquid nitrogen. Then the female reproductive structure. So gynoecium, that includes the group of pistils. Pistil means which include the stigma, style and the ovary. Alva, stigma, style, ovary idre adanna pistil anta kariteve alva. So here, sometime the gynoecium may consist single pistil, that is called as monocarpellary. So here only single stigma, style and ovary is called is present, then it is called as the monocarpellary. When more than one pistils are present and they are fused. So if they are fused, that is called as the syncarpous condition, or if they are free, that is called as the apocarpus. What is syncarpus? Syncarpus means pistils are more but they are fused. Apocarpus means they are free and uh, more than one pistils are present but they are free, that is the apocarpus condition. So here examples for this given in the diagram, that is very important: apocarpus and the syncarpus ovary. Okay, then the megasporangium. So here, this is the ovary. Inside the ovary, ovules are located, right? The ovules are present inside the ovary, and these ovules, they contain the embryo sac and the female egg. Female gamete is present inside the ovule. So here the ovule mainly, that is, yes, that is also called as megasporangium. So ovules that is present in the ovary, and that is also called as megasporangium. So this is the structure of the ovary. And funicle, so it is place where it is attached, that is the stack like structure, helps to attach the ovary. And the hilum, this is the hilum, and two integuments are present, that is protective in nature. And the micropyle end, so this is the micropyle end where the integuments are absent, and this is the chalazal end. So this is the structure of the ovule, which contain the embryo sac inside it. Okay, so which contain the embryo sac inside the ovule. So megasporogenesis, how the female gamete is formed. Here also in megasporogenesis, the some cells that is present in the nucellus. So here inside the ovule, some nucellus cells are there, so they become megaspore mother cell. So before the formation of the embryo sac, some nuclear cells, they become megaspore mother cell. Megaspore mother cell means that is also diploid in condition, that undergo the meiosis and form the 4 megaspores. Same as microspores. Megaspores are produced, but out of 4, 3 will degenerate and only one become functional. So in case of megasporogenesis, 4 megaspores are produced, but only 3 remains, and 3 degenerate, only 1 remains. The remaining one, so here microspore mother cell meiosis to undergo the, sorry, this is the microsporogenesis video. Here the only one functional megaspore, the functional megaspore that will develop into a female gametophyte. So the functional megaspore that will develop into female gametophyte, that is the embryo sac. Okay. So here from one megaspore mother cell, embryo sac will develop. So here this method is called as monosporic formation of the uh, monosporic development, because single megaspore is developing the embryo sac. So it is called as the monosporic development of the embryo sac. Here after the formation of the embryo sac, so there is, this is the functional megaspore which undergo the mitotic division continuously and form the 8-celled stage. So functional megaspore that undergo the, this is the megaspore within the embryo sac and form the 8-celled stage. After the 8-celled stage, there is an arrangement of the cells takes place that form the mature embryo sac. Here this mature embryo sac, that is called as 7-celled and 8-nucleate stage. 7-celled means here 3 cells at the chalagal end. In out of 8 cells, 3 cells at the chalagal end, and here 2 synergid cells and 1 egg cell. So here also 3 cells, 6, but at the center here 2 cells. So here single cell is present, but it fuses and form the, here at the center, the central cell is formed by fusion of the cytoplasm of the 2 cells, but 2 nucleus is present at the center. 2 nucleus, but only 1 cell is present, that is the central cell. Okay, that contain the 2 nucleus. So this is why, this is what the 3 plus 3, 6, 7 celled and the 8 nucleate. Nucleus is 8, but the 7 celled. So this is the mature embryo sac diagram, how it forms the mature embryo sac. Then already male and female gametes are formed, but there should be a transfer of the pollen grain to the female gamete, right? Pollination, so that helps for the transfer of the pollen grains to the stigma of the female reproductive structure. Here autogamy, what is the definition of the autogamy? You should know this definition, so this will come in MCQ question. Autogamy means the pollination is achieved within the same plant. Transfer of the pollen grains to the stigma of the same flower, that is called as the autogamy. Here chasmogamous flowers and the clistogamous flowers. So chasmogamous flowers, so which are similar to the flowers other species with exposed stigma and the anther. Chasmogamous flowers means which has the exposed stigma and anther. Clistogamous flowers means which do not open at all. So they perform the autogamy. So they do not open at all. Within the flower only, within the closed flower only, they'll pollinate. That is the clistogamous flowers. Example for this, Viola oxalis and commonina produces two types of the flowers, both chasmogamous and the clistogamous flowers. That is very important. And next one, geitonogamy. Transfer of the pollen grains from the anther to the stigma of the another flower of the same plant. So that is the geitonogamy. Xenogamy, that we can see in papaya. Here the transfer of the pollen grains to the stigma of the another flower in the another plant. Okay, so that is called as the xenogamy. So here flowers are unisexual flowers, means which contain either male or female reproductive structure. Then agents of pollination means some biotic and abiotic agents are involved in the pollination. Here some abiotic agents, that is the wind. Here, what are the characteristics of wind pollinated flowers? That is very important. So here the pollination requires the pollen grains light and non-sticky. The pollen grains are light and non-sticky. They can be transported in the wind current. They possess well exposed stamens. Here in case of the wind pollinated flowers, they have the well exposed stamens. Why? So the pollen grains that should be easily carried with the wind current. Then large feathery stigma easily trap the airborne pollen grains. Then one more, wind pollinated flowers often have single ovule, each ovary and numerous flowers are packed into an inflorescence. So here for example, corn cob, the tassels you see nothing but the stigma and the style. So in case of the corn cob, the tassels what you see, that is the stigma and the style which trap the pollen grains. So this is the very important characteristic feature of the pollination by wind, that is the wind pollinated flowers, important for your MCQ CET competitive exam. Then pollinated by water. Here very less plants they pollinate by water. For example, only 30 genera. Here what are the characteristics of the water pollinated plants? Here some examples for water pollinated plants, Vallisneria and Hydrilla, so which grow in fresh water, and several marine sea grasses such as Jostera. So they show the water pollination. See here, not all aquatic plants use the water for pollination. The majority of the aquatic plants are water, hyacinth, water lily. The flowers emerge above the level of water and pollinated by the wind. Here, water hyacinth and water lily, they are living in water, but uh, they pollinated by wind or the insect. Okay, so they are not pollinated by the flowers are not pollinated by the water current. Here see here very important example. In Vallisneria, the female flower reach the surface of water by the long stalk, and male flowers or the pollen grains release onto the surface of the water. So this is the female flower. This is the female flower, and the pollen grains released male gametophyte that is released on the surface of the water and that reached the stigma. In another group of plants such as sea grasses, female flowers remain submerged in the water. Female flower is that is inside the water. So the pollen grains in species long and ribbon like, and they carried passively inside the water. This statement is very important. So in case of sea grasses, pollen grains are long ribbon-like and they are carried passively inside the water. And the pollen grains have the mucilaginous covering that protect it from the wetting. Okay, so these are the features of the water pollinated flowers. Very, very important one. Pollination by insects. So insect pollinated plants. So here not only insects, lemurs, tree dwellings, rodents and many other organisms, they also helps in the process of the in pollination. Here these insects, they pollinate the flower as well as a reward they get the nectar from the flower. So here insect pollinated flowers are large, colorful, fragrant and rich in nectar. Okay, so that is the very important point. And here nectar and pollen grains are the usual reward for the insects. So the insect, they are attracted by nectar uh, and they eat some pollen grain also. Some animals also, they perform the pollination. Here, in some species, floral rewards are providing safe places to lay the eggs. For example, the tallest flower, Amorphophallus, very important. The tallest flower, Amorphophallus. So that is the flower that is about 6 feet height, which provide the safe place to lay the eggs for the insects. Okay, then similar relationship that found in between yucca and the species of moth and plant. Here the moth deposits the eggs in the locule of the ovary and the flower in turn gets pollinated by the moth. Moth lay the eggs inside the locule of the ovary, and the flower that is pollinated by the moth. So here the larvae of the moth come out of the eggs and seeds start developing. So the larvae that come out from the ovary when the seeds start developing. So this is how the mutual relationship that found in between the moth and the flower. Okay, so this is the very important one. Then outbreeding devices, how the plants are able to they are able to they what you call these plants. So they produce the some different types of the flowers, means they are encouraging the, yes, so the exact word is they are encouraging the outbreeding devices, means they are encouraging the cross pollination and they discouraging the inbreeding. So they are discouraging the autogamy. So why they are giving the more importance, why the flowers are encouraging the cross pollination? So why not the self pollination? So this self pollination that leads to the inbreeding depression, right? So the flowers itself, they developed some mechanism to encourage the cross pollination and also prevent the autogamy. Okay, here some anther and stigma are placed at the different position, and there is no synchronization of the release of pollen grains and the receptivity of the stigma. And sometime by producing the bisexual flowers. So these all are the outbreeding devices. Here stigma and style that is at the same different position cannot come in contact with the stigma of the same flower. Both of these devices prevent the autogamy but not the gytonogamy. So here autogamy is prevented, but gytonogamy is not prevented. But in some cases, some flowers, they produce the unisexual flowers. So to prevent the cell pollination is prevented in the unisexual flowers. So here in this cases, autogamy as well as gytonogamy is, so that is here it prevents the autogamy but not the gytonogamy. So if both male and female flowers are at the same plant, such as castor, so male and female flowers are at the same plant, so that can prevent the autogamy, not the gytonogamy. So gytonogamy that occurs because within the same plant there is a chance of the pollination. In several species such as papaya, male and female flowers at the different plant itself. So it is preventing the both autogamy and the gytonogamy. Male flower is in one plant and the female flower is in another plant. So there is no autogamy and the gytonogamy. There is only xenogamy takes place. Okay, that is in case of the papaya. Then the pollen pistil interaction. So the pollen grains, when it reach the particular stigma, so when it reach the stigma of the same species or the compatible stigma, then only it is able to germinate. Otherwise it is rejected by the stigma by releasing the some chemicals. Okay, so that is called as the pollen pistil interaction. Here pollen grains, when they reach the particular, the correct stigma, then only they are able to germinate. Otherwise if they reach the different species stigma, they are not able to germinate. Here during the germination, the pollen tube that is formed, and this pollen tube that grows towards the micropyle end of the embryo sac, and which releases the two male gametes, the generative cell which releases the two male gametes and which reaches the micropylar end of the embryo sac. Artificial hybridization. So here mainly artificial hybridization that is carried out to increase the productivity, to get the desirable characters. So it is performed by two steps. First step, that is the emasculation, where the male anthers are removed, that is called as the emasculation, and bagging. So emasculation that is followed by bagging. So bagging that prevents the unwanted or the contamination of the stigma. When you get the, or when the stigma become receptive, then the pollen grains collected and tapped on the stigma and rebagged. So the bagging process that prevent the stigma from the contamination. So that is the two main steps of the artificial hybridization. Then the double fertilization. So the two male gametes that is released by the generative cell. Here the one male gamete that fuses with the egg cell that is present at the micropylar end, right? So here the egg cell is present, one male gamete that fuses with the egg cell, and second male gamete that fuses with the central cell, so which is already in the 2n condition. So the central cell that is already in the 2n condition, and one more male gamete fuses it with this and which form the 3n condition, that is called as PEN, primary endosperm nucleus. Okay. So this is called as the double fertilization. After the fertilization, these antipodals and the synergids, so these all um, degenerate. Only zygote and the primary endosperm nucleus that remains. Here post fertilization events. Here post-fertilization events that includes the formation of the maturation of ovule into seed and ovary into fruit. So these all things are involved in the post-fertilization events. The very important thing is the endosperm. So the primary endosperm nucleus that develop into endosperm, which provide the nutrients to the developing embryo, zygote that develop into embryo, right? Endosperm providing the nutrition to this developing embryo. Here, this endosperm that is present in the seed. So some seeds or the some embryo, they consumed the endosperm completely during the development. That is called as the non-endospermous seeds, which do not contain the endosperm after the development. But some seeds, they contain the endosperm after the development of the embryo also. That is called as the endospermic seeds. Okay. For example, in case of the castor and coconut, endosperm is present. But here, in case of some peanut, pea, and groundnut, endosperm is completely consumed by the embryo. So that is called as the non-endospermic seeds. And here, the embryo that the development of the embryo from the zygote that involves the same step like globular, heart-shaped, and the mature embryo in case of both dicot embryo and the monocot embryo. So this is the structure of the dicot embryo and this is the structure of the monocot embryo. So this is the developmental stage of the monocot embryo. And the very important thing, seed. Here the seed that is the fertilized ovule, right? Fertilized ovule is called as the seed. Seeds are two types, non-albuminous and the albuminous. Already I explained where the endosperm is completely consumed. That is the non-albuminous. Albuminous means endosperm is present. Some seeds such as black pepper, beet and remnants of the nucellus are also present. Nucellus is also present that will develop into perisperm. Nucellus that develop into perisperm. And a very important thing here, some seeds, so they show the dormant. So when they get the water, then only when they get suitable condition, then only they are able to germinate. Here some seeds, they show the dormancy many years, around 10,000 years of dormancy also they showed. Okay. So that depends on the dormancy of the seed, that depends on the how the oxygen or availability of the water and the suitable environment for the germination. Then the fruits, that is the fertilized ovary, right? Fertilized ovary, that is called as the fruit. So here some fruits, they develop without fertilization, that is called as the parthenocarpic fruits. Here for example, banana. And here, as I told, some seeds, they show the, they found, uh, so oldest seed, that is the Lupinus arcticus. So this is the oldest seed from the Lupinus arcticus that is excavated from the Arctic tundra. So that the seed germinated after 10,000 years of the dormancy. And one more, Phoenix dactifera, that is the date palm that has the dormancy of the 2,000 years. So that is found near the King's Herod's face palace. So these two are very important, Lupinus arcticus and Phoenix dactylifera, which shows the very high period of the dormancy. Then the apomixis and polyembryony. Examples and the definitions. Apomixis means it is the formation of the seed without fertilization, or the seeds are produced without fertilization, that is called as the apomixis. Here, the species of Asteraceae and grasses have evolved to the special mechanism. So that is without to produce the seeds without fertilization called apomixis. So examples Asteraceae and the grasses. Very important. Note down this. And so that is the apomixis and the polyembryony. Here, what is the definition of the polyembryony? In some species, so here citrus and mango varieties, some nuclear cells surrounding the embryo start dividing and protrude into embryo. So not only one embryo, within the single embryo sac, many embryos will develop, that is called as the polyembryony, because nuclear cells, they develop into a embryo. So the embryo sac and they develop into the embryos. So here, for example, citrus and mango. So that shows the polyembryony, citrus and the mango. Or occurrence of more than one embryo in a seed, that is called as the polyembryony. So more embryos are developed, the nuclear cells that is also developed into embryo within the seed, that is called as the polyembryony. So this is the brief explanation about the sexual reproduction in the flowering plants, because already you learned, I hope you learned this topic. So this is just a recall of this chapter. Okay. So let us solve the some MCQ questions related to this chapter. First question. Yes, we are going to solve some important questions, MCQ questions from this chapter. I hope all are ready to solve the question. Read the first question. Flowers with both androecium and the gynoecium are called what type of flowers? Bisexual, anther, stamens, and the unisexual. Yes, they are called as bisexual flowers, or they are called one more word, monoecious. Monoecious flowers. Monoecious. Monoecious flowers means mono means within the single flower, male and female reproductive structures are present. So that is called as the monoecious condition. Anther, that is the male reproductive organ structure, and the stamens which contain the anther and filament. Unisexual means that is the that contains single male or female reproductive structure, any one. Second one, the transfer of pollen from anther to the stigma is called. The transfer of the pollen from anther to the stigma is called pollination, fertilization, adoption, diffusion. So very easy question. That is called as the pollination. Third question. The fusion of female reproductive nucleus with the male reproductive nucleus is now adoption, excretion, fertilization, regeneration. So these all are very basic level question. The fusion of female reproductive nucleus with the male reproductive nucleus is known as, yes, what is the answer? Adoption, and the excretion, that is not related to this. So this is called as fertilization. The correct answer, that is 3, fertilization. Next question. The two nuclei at the end of the pollen tube are called tube nucleus and generative nucleus, sperm and ovum, generative nucleus and stigma, tube nucleus and the sperm. Here, two nuclei at the end of the pollen tube. So here the pollen tube that is germinated. Just a minute. So the pollen tube that is formed on the stigma, and at the end of the pollen tube are called tube nucleus and the generative nucleus. Sperm and ovum? No, sperm and ovum is not correct answer. Generative nucleus and the stigma? No, that is the correct wrong answer. Tube nucleus and the sperm. So that is also wrong one. Here the tube nucleus and the generative nucleus. So that is called as two nuclei are called as tube nuclei and the generative nucleus. Next question. Generative nucleus divides forming two male nuclei, three male nuclei, two female nuclei, three female nuclei. The generative cell that forms the male gametes, right? How many male gametes it produces? Yes, generative cell that produces the two male nuclei. So the option 1 is the correct answer. Generative nucleus that divides and forming the two male nucleus. Next question. Embryo sac is located inside the stigma, ovule, micropyle and the style. Embryo sac is located inside the what? Yes, embryo sac is located inside the ovule. Next question. One nucleus of the pollen tube and the secondary nucleus of the ovum grow into stigma, endosperm, anther, stamen. Yes, what's the answer for this question? Yes, what's the answer? C. No, anther is not a correct answer. Here, one nucleus of the pollen tube. So here the generative cell that produces the two gametes. So in that one nucleus of the pollen tube means, so the one male gamete and the secondary nucleus of the ovum. So here this is the secondary nucleus of the ovum and one tube nucleus that fuses and form the which of this following structure? That is the endosperm. Next question. The stalk of datura flower at its base it is known as pedicel, corolla, sepal and the thalamus. Here the stalk of the, just imagine this is the stalk of the flower. Yes. So this just imagine this is the petal. Okay. So corolla that is the colorful part and sepal that is the greenish color part. The stalk of the datura. So this stalk what it is called? That is called as pedicel. What is thalamus? This is the bulged portion from where all the petals and sepals that arise, that is called as the thalamus. So here the correct answer that is the pedicel. Next question. The male reproductive parts of the flower, the Stamens are collectively known as androecium, filament, anther, gynoecium. The group of stamens, that is the male reproductive structure, that is collectively called as, so the very important part, that is the androecium. Then the other name of gynoecium is pistil, stigma, androecium, style. The other name of gynoecium is, yes, what's the answer? That is B. Is it stigma? The other name of the gynoecium, that is the pistil. Stigma means only receptive part. Pistil means stigma, style and ovary. Together it is called as the pistil. So that is the group of pistils that is known as the gynoecium. Okay. Yes, so these are the few questions related to this chapter. In the next class, we are going to solve few more questions on this chapter. I hope this session is useful for you people. Thank you, dear students. We'll meet in the next class. If you have any doubts, please comment in the comment box. Okay, thank you.